But hello, everyone, and thank you for joining us today. We are truly looking forward to today's Empowering MBC webinar. And for those of you that are new to the series, my name is Lauren Jost. I'm the project coordinator for the breast cancer program here at Washington University. And I'm the main point of contact for the webinar series, as well as our Living Empowered MBC support group. If you have any questions or suggestions or would like to get more involved, please don't hesitate to reach out to us at our email at empoweringmbc@wustl.edu, of which I will put that in the chat for easier access as well. I will also put our Siteman webpage in the chat to make life easier for everybody so they can get as much information as possible. Here we do have with us today our two lovely breast oncologists with WashU and Siteman. We have Dr. Nuseva Bagegni, who is an associate professor of medicine, our associate medical director of clinical research in the Division of Oncology, and the leader of our breast cancer clinical trials portfolio. We also have Dr. Cynthia Ma, professor of medicine, and our clinical director here at WashU for the breast program, both who currently are leading numerous breast cancer trials, both in early stage and advanced stage breast cancer here at WashU and Siteman. And together, they're going to share their expertise during today's session of Advancing MBC Care Clinical Trials 101. Give me just one moment. I'm going to admit a few more individuals. I'm multi-screening today, so we're all over. I would also like to say that we're thrilled to welcome Josh Newby, the founder and executive director of Teresa's Research Foundation, who is one of our partners in our webinar series, as well as our support group. He is going to moderate our town hall discussion, which will be theoretically the last 30 to 40 minutes of today's time frame for the webinar. And we thank you, Josh, for being here and joining us today. If you have any questions during today's presentation or comments, please feel free to put them in the Zoom chat, and Josh will do his best to get to them as soon as he can. This webinar is being recorded, which I think you all got a notification for. I will cut the recording once the town hall starts, and these will be posted on our Siteman webpage within the next week or so for anyone that's interested in either rewatching or sharing. Do note that this webinar is informational use only. So if you have any questions about your personal care or any trials that you feel that you would be interested in learning more about, please ask your provider. And finally, after today's session, there is a short anonymous Zoom survey that will pop up in your browser once we sign off of Zoom. We would truly appreciate it if you could take just a few minutes. It's only 8 questions long to give us feedback. Any feedback we get for these webinars is super beneficial, and we want to be able to further the education and experience that everyone has with these webinars and the MBC community here at WashU and nationwide. With that, I'd like to hand it over to Dr. Bagegni, who is going to start our presentation. Thank you so much.
Okay, wonderful. Thank you. So thanks so much, Lauren, for the introduction, and thanks for everybody for joining us today. So we will talk about advancing metastatic breast cancer care, and we're going to talk about clinical trials 101. So the first part, I'll talk about a little bit about what clinical trials are, the purpose, the goals of trials, a little bit about essentially like drug development and how trials happen and the different phases. And then Dr. Ma will talk about some novel therapies that are in development. Before I forget, I did want to mention that we do have a webinar coming up. We're coordinating with our cardio-oncology Center of Excellence along with PRG to provide some information about cardiotoxicity of breast cancer treatment. So that should be coming, and then Lauren will be announcing that in the newsletter as well once that information comes forth. So I just wanted to throw that out. So we are just on the heels of the San Antonio Breast Cancer Symposium. So many of us were just in San Antonio last week at this really important meeting. It's the largest breast cancer research meeting in the world, and it's really where clinical providers, researchers, investigators, basic scientists, patients, patient advocates join and come together to talk about the latest data, either preclinically prior to going into the clinic, or new drugs and new trials and data from trials. And so I think it's really important for us to recognize that. And this is actually why we selected today as the time that we're going to discuss clinical trials. I've had a number of you ask me previously, how can we get involved? They do have registration for patients and patient advocates. And so if you log on to San Antonio Breast Cancer Symposium, the program, they don't have it up for next year yet, but you can look and see, and they do have a discounted registration fee, and they do have different scholarships and so on to help with that. So what are we going to discuss in part one? We're going to review the purpose of clinical trials, the various phases and their goals. We'll briefly walk through the process of drug development, FDA approval. We'll review key parts of participating in a clinical trial, some common misconceptions. We're going to discuss inclusivity because this is really, really critical. And we're going to discuss some of the evolution of how we design clinical trials, and many are actually patient-driven. So some great resources that I'd like to reference here are from the MBC Alliance and breastcancer.org. And so I thought that this is really, really important. So breast cancer tops what we call the most studied diseases list. And so if you see in 2021, 2022, 23, 24, you can see breast cancer tops the list. And then we had COVID-19, with the pandemic, and then other types of diseases. And so we've seen a significant evolution in approvals for metastatic breast cancer. And you can see here back in the 70s, actually, the first targeted therapy that we had was tamoxifen. So many of you may be familiar with this. This is an anti-estrogen therapy, and that was approved in the late 70s. And then we had various hormonal therapies that got approved. And just over the past couple of years, we've had additional more novel hormonal therapies. But early on, we had a lot of chemotherapy that was being approved, and chemotherapy, very kind of general type therapies. But as we learn more about the biology of breast cancer, we really understood what drives growth of the breast cancer, and we've been able to really pinpoint that and develop very targeted therapies. So in late 90s, 1998, we had Herceptin or trastuzumab. This is a treatment that we use for HER2-positive breast cancer. And then we had other types of novel therapies. We call them antibody drug conjugates. So they're smarter ways of giving chemotherapy to minimize side effects. And then we have these CDK4/6 inhibitors, which are like Ibrance and Kisqali and Verzenio, and then a lot of other targeted therapies. And then we have immunotherapy like pembrolizumab that's approved for triple-negative breast cancer in certain situations. So you can see that we've really evolved from a chemotherapy focus to a shift of targeted therapies that really are getting to the underpinnings of the biology of breast cancer. So more to come in part two of this webinar. Dr. Ma will talk through what's to come beyond 2025 as we go into 2026. And so significant treatment achievements have been made, and clinical trials really allow us to have development of novel, more effective, and safer therapies and really shift the status quo. It shifts the standard of care. And I like to think of it as like pushing the envelope, right? You have this person that's in the envelope and they're trying to push out. So the ultimate goal of clinical trials is really to allow for more and better treatment options and ultimately improve survival and improve quality of life. And so I really like this picture here. So everybody has to come to the table. We have what we call stakeholders. And, the stakeholders are patients, so people who are going through the diagnosis, physicians and investigators, industry and companies that make therapies, and then the FDA. And so you have all these stakeholders, and the aim is for better outcomes, better treatments, safer treatments, and all of that has to align to really see progress and make progress. So everybody has to come together, sit at the table to really advance metastatic breast cancer care, and everybody has a piece to include in the puzzle. So what are clinical trials? So clinical trials, sometimes we call them studies. They're really conducted with a goal to improve medical care over time. And as we learn more about the biology, or we learn more about different types of treatments and the side effects, and then what happens after the cancers see those treatments? How do cancers rewire after treatments? We're learning more about how they become stubborn to it, for example. Then we can improve on that and learn more and more. And that's why many of these clinical trials do require various like blood samples and biopsies, because what we're trying to do is we're trying to understand more about how the treatment works and potentially how it could stop working over time so we could develop better treatments and hopefully eventually a cure. And this really relies on volunteers to participate. And so participating in a clinical trial is, it's completely voluntary. Nobody should force you. And your doctor would be talking to you about standard options, but also potentially enrolling in a clinical trial if it's the right thing for you. So it really relies on volunteers to participate. And there's various different types of clinical trials, and we'll talk a little bit about broad categories. And every FDA approved, or essentially nearly every FDA approved therapy available to us today has gone through a very careful, robust, lengthy evaluation process. And we're going to discuss this shortly. So clinical trials can be conducted for various reasons. So number one, to study new ways of diagnosing a cancer or monitoring the treatment response. So for example, at Siteman, we have a clinical trial where we're trying to see if we could use a blood test to help determine how the cancer is doing rather than doing scans all the time. We know that scans can come with a lot of anxiety. We have a term for it. It's called scanxiety. You can feel fine, and then the day before your scan, then it's like, you know, just a nightmare, right? So we all acknowledge that. So are there better ways of really diagnosing the cancer, monitoring how the cancer is responding to treatment, even better ways of doing scans? So we have these PET scans. We have very sophisticated types of scans that look at estrogen based breast cancers. Actually, that was developed here at Siteman and WashU. So it could be really from a diagnostic perspective. The other, which is a lot of what Dr. Ma and I do, is testing new medications and new ways to use existing medications, perhaps in combination for cancer treatment, and really trying to understand, you know, what are different tests or tools that we can use to help determine what's the right treatment for the right individual. And then making sure that therapies are safe and that we understand the safety profile and we know, you know, what do we need to be on the lookout for with certain medications. And then also not the least, developing strategies to improve quality of life for people living with cancer. So we've had a number of drugs that have gotten approved, and we realize this is incredibly toxic, causes a lot of diarrhea. And if we start at a lower dose and then titrate it up very, slowly, then patients can continue on with treatment. So ultimately, a treatment won't help us if we're not able to continue it. And so this is really important. And we also want to make sure that the treatment isn't, really what's impacting our quality of life. So two major types of clinical trials. We have therapeutic trials and we have non-therapeutic trials. And therapeutic trials, these are, again, investigating a new treatment, its side effects, how effective it is, and the questions here we're asking is the new treatment safe? And are the benefits of the new treatment or the new procedure or the new test, are they going to outweigh the potential side effects? And as we're moving along more and more, we are incorporating patient feedback. And these are what us as oncologists and researchers call PROs, or patient-reported outcomes. And so if any of you have participated in a clinical trial, you may be asked to complete a number of surveys. I think sometimes they can be somewhat burdensome, but it is important to understand how participants in clinical trials are feeling. What's their quality of life? How do they feel that treatment or that what's being investigated is impacting their life? And then the other is, does the new treatment work? Is the new treatment going to be better than the standard treatment? And does it work in a group of people who did not see an improvement in the standard treatment in the past? And so that's therapeutic. And then we have non-therapeutic, which is that diagnostic imaging type trial, kind of what I mentioned, special types of PET scans or MRIs, and other supportive care and symptom management trials. And these are oftentimes called quality of life trials. So studying ways to reduce treatment side effects, improve overall functioning and quality of life, and these can be, different ways of giving a treatment perhaps, or also looking at lifestyle related studies, including diet and exercise, or other ways of monitoring for side effects as well. So this is a really busy slide, but this is just to show that this is a pretty long process of drug development. And here we're talking about going from the lab to FDA approval. And so the first phase is the discovery phase, and this is what we call preclinical. So this is before it goes into the clinic, into humans. There's a lot of studies that happen in the lab, and it's important to determine if there's a biologic underpinning to the cancer. We're understanding that, and then we're testing a medication, and then it has to be tested on different cells and different animal models, usually at least two, and beyond that, developing the right dose and so on, so that it could then move forward into testing in humans and people. And at this point, not anybody can just come and say, okay, we're going to start doing this in humans. It has to be formally submitted to the FDA. The FDA has to approve, review it diligently, approve it. A lot of times they come back and give additional feedback, and then it has to be reevaluated. And so this is called an IND application. So the drug is now approved for testing in humans. And then we have various phases, including phase 1, phase 2, phase 3, and this is what we call the clinical trial phase. So this is usually when we talk about clinical trials, we're kind of focusing on one of these phases. And then we go to FDA approval, and then there's a phase 4, which we'll talk about. So this is that discovery phase, preclinical. So first you're testing these new treatments to show promise in the lab or in animal models. And then these treatments may ultimately be tested in clinical trials involving humans if there is a signal that it may be beneficial. And we also have to have some safety measures that are identified to really select the right dose. And that's why it's tested in animals as well. So I think this is a little bit easier to follow. Phase 1, the question here is the treatment safe? And here we're trying to understand the right dose, and we'll get to each of these phases in a little bit more detail. And then phase 2, we're trying to say, does the treatment work? We're testing it in more humans and learning more about the drug. And then phase 3 here, we're trying to say, compared to the standard treatment, is this new treatment better? Is it more effective? Do patients do better with it? Do they have less side effects? And then it gets approved. And then we have phase 4, which is now it's available for prescribing, and they call it post-marketing surveillance trials. So now it's available. Now it's being given to many people, and we're able to now capture potentially new, more rare side effects. So even with some of the weight loss medications that are available, now that it's widely available for most people, and we have a lot of people that are on these therapies, we're learning more about these side effects that maybe weren't necessarily seen in the clinical trials. And so in oncology and in cancer trials, we really start to look at real world evidence. So we're seeing more of that now that we have more databases. We're able to go through an insurance database and say, for these individuals that got this treatment, how have they been doing with it? How long were they prescribed that medication? And so sometimes this could help us understand better in what we call kind of the real world, how individuals are doing with the treatment. So after it goes through this clinical trial phases, then the FDA has to go back. So whoever is developing that therapy has to go back and submit another application. It's called a new drug application, and the FDA has to review it, and they have to meet certain measures. And usually it happens throughout the course of this clinical trial phase. And then if it's looking promising and so on, depending on the duration, then the FDA grants approval. And that's when we go to this phase 4, which the treatment is available. And now we're looking at long-term safety in a larger group. So this takes a long time. This is about 10 to 15 year period from start to approval. So just kind of recognizing the treatments that we've been through, many of them have gone through this very, very rigorous evaluation phase. So what is a phase 1 study? So here what we're doing, and when I've explained it, to my patients, here we're trying to understand, there's a therapy that appears to be beneficial, or it's a combination of treatments that perhaps is approved for another type of cancer. And here we're trying to understand what's the right dose and is this safe? So that's really the primary goal. And so it really requires a lot of follow-up, and it usually requires a lot of testing and visits. And we'll talk a little bit about why we do that testing. Here, the goal is we want to optimize effectiveness and we want to minimize side effects. So it's really trying to strike that balance. Again, this is the first time we're evaluating a new cancer treatment with the objective of treating the cancer. So even though it's a phase 1 study and we're just learning about it, and the focus here is this safe? What's the right dose to move it forward? The objective is also to treat the cancer. So if your doctor is talking to you about this, then it's also the treatment for your cancer. So here we're defining safety, we're identifying side effects, and we're identifying the right dose that's going to move forward. And then there's something called PK assessment. So what does this mean? So why, if we're enrolling in a phase 1 study, why are we having patients come in so frequently or get so many lab draws? So sometimes we have patients coming in on the first day, and then a day after, and then they're not next week, and then the day after that, and then the following week. So the reason why is we're trying to understand how the body is affecting the drug. And so for those who have participated in these studies, we have them come in and we do a lab, then we have them take the medication or get the infusion, and then maybe 30 minutes later, an hour later, 2 hours later, 4 hours later, we're checking the blood levels because we're trying to understand more about absorption of the medication, how it gets distributed throughout the body, how it gets metabolized and cleared through the body as well. So it's essentially the movement of the drug in and through and out of the body. And so this is why phase 1 studies, because we're trying to understand what's the right dose, the safety. It requires extensive monitoring. And so it's something that usually requires more of a time commitment. So what about phase 2? Here, we've understood a bit about safety, but we're always looking at, the safety profile of the medication. And then here we're really focusing on the dose that we've selected. We want to see, the effectiveness in a larger group of patients. And so these trials tend to be much larger. And here the focus again is understanding more the efficacy in a larger group. So this is the most common phase where drug development actually fails. And in clinical trials and research, we call it go or no go. And so this may help to guide if we're seeing that signal and we're seeing safety, then we move it forward. And here we're really gaining experience with how to give the medication, and how to support patients through the side effects as well. And then for a phase 3 trial here, we're saying, can we redefine the standard treatment? So we used to give this treatment. Now we think that this treatment is going to be better because of all of these reasons and everything that we've seen with phase 1 and phase 2. And so these types of studies are done in hundreds to thousands of patients. They're larger. They oftentimes are across either the entire country or usually globally. And here you're trying to compare the new treatment versus the standard and wanting to prove that it's better, or it may be more effective or safer for patients. Or you might say everybody gets the standard treatment, but we're going to add something to it that may allow for a better treatment response versus a placebo in addition. We're going to talk a little bit about placebo because I think this is one of those common misconceptions. But these are really the trials that are going to eventually lead potentially to an FDA approval. And clinical trials through all of these phases, there's a lot of oversight. So I usually tell my patients, if you're in a clinical trial, it's not just us, your medical team seeing you, but there's a huge team behind you that's taking extra, there's extra pairs of eyes and ears, and there's an extra radiologist that's reviewing things, and people that you may see and may not see. So most clinical trials must be reviewed by various scientific committees along the way, including what we call an institutional review board or an IRB. So this is a committee that reviews research to make sure that the rights of patients are protected, that their safety is protected both before the study starts and as it proceeds. And it's not just a one-time thing. It happens all the time. And the study team that's conducting the study, they're supposed to check back in periodically to provide any additional updates, and that's all mandatory. So if there are any major updates, the IRB must be informed and things must be approved to move forward. And so what are some of the questions that the IRB will ask the research team? They're going to say, is this study being conducted ethically based off of what's currently available? Is this, the right approach to take? And it really, it includes a very multidisciplinary group. They can be physicians and patient advocates. So it tends to be a very diverse group of individuals that come together and make the committee. Are proper safety measures and monitoring in place? Are patients adequately informed of the current state of the study? If it's a phase 1 study and we know that we could potentially see certain side effects, all of that has to be told to participants. What we know and what we don't know, especially in those very early studies, and that's through what we call an informed consent. So I mentioned this previously, but participating in clinical trial is voluntary. It is important that people understand that you're not to be forced to participate in a clinical trial. But what I tend to think of it is it's another tool in the toolbox, and it's another opportunity potentially as a treatment to treat the cancer. And this informed consent form, it tends to be not just a few pages, it tends to be a pretty hefty document, and it's provided to all participants to review beforehand and before they're being considered for participating in the study. And this is going to detail the trial specifics. So why the trial is being conducted, who's eligible to participate, what's known about the treatment, what are possible risks and benefits to participating, what alternatives do you have available. They won't necessarily list that out, but they'll say, there may be other alternatives you need to talk to your doctor about. What's the duration of the trial? Is it, a few months? Is it years? And then any testing procedures that will take place and who will pay for that? What are the costs, including time, including lodging, gas, and mileage, and things like that. So giving written consent does not automatically mean that somebody will be accepted in the trial, and informed consent does not stop at the start of participation. So if there's any updates, then you will be connected by, the research team to say, okay, we've had some additional updates and we have to go over that and rediscuss. So informed consent, I always say it's not a one-time thing, it's a process. So inclusivity matters. So this is really, really important. So diversity in clinical trial participation drives improved outcomes. So many of us either take or have know people who take blood pressure medication or other types, cholesterol medications. All of those medications have gone through clinical trials. So we've all been impacted by clinical trials. But a lot of times we have significant underrepresentation in clinical trial participation. So if we don't have a participant population in the study that is representative of our community, then we may not really understand how effective a certain treatment will be in a diverse group of people. So among 32,000 individuals who participated in new drug trials in the US in 2020, only 8% were Black, 6% Asian, 11% Hispanic, and 30% ages 65 and older. So you can see that there's a significant disparity here. And these estimates have worsened since 2019 and are not really aligned with the US Census data. And so back in the 90s, in 93, the US Congress passed the NIH Revitalization Act. And this really called for increased inclusion of women and racial and ethnic minority groups in all federally funded clinical research to really ensure the generalizability of results within these populations. And we did see that there was an increase in clinical trial enrollment, yet the proportion of minority patients in these trials actually declined. And in 2014, only about 1% of the National Cancer Institute sponsored clinical trials were primarily focused on racial and ethnic minorities. And we know in breast cancer trials, usually the minority populations are underrepresented quite significantly, so far less than 10%. So having this underrepresentation is going to really lead to persistent gaps between trial and representation and really the real world burden of certain diagnosis like breast cancer. We're going to have limited confidence in treatments that eventually become approved and how it may lead to unforeseen consequences. And we know even from, women and men, or in terms of certain medications for even cholesterol or diabetes or whatnot, that metabolism can vary and outcomes can vary. So inclusion and representation is really important. And, if it's not there, then we may actually have worse outcomes, and we can potentially not understand that there's less effective or even harmful side effects to certain groups of people. So many factors have really contributed to this. So one of which is lack of awareness or access. So some people say, I just didn't realize that I can participate in a clinical trial. I think another big thing is historical mistrust in the healthcare system. And so, just historically, we've had some situations that have happened. And so and that's what actually led to instilling, things like all of these scientific and ethical committees like the IRB and so on, to make sure that we uphold, ethical research and, patients' autonomy and participants' autonomy and safety. And then others are socioeconomic factors. It does take some time, to participate in these studies because of the various testing that goes along with this. The other is fear and misconceptions, and we'll talk a little bit about some of these misconceptions. And then restrictive inclusion criteria. So this is a big one that Dr. Ma and I it bothers us. So patients who have bone-only metastatic breast cancer, can be a tough one. For brain metastasis. So sometimes studies will exclude patients with that. So that's another one. Lobular breast cancer sometimes can be difficult to identify on imaging. So we need better strategies for that, but sometimes that can be a problem as well. So again, we really need inclusion. We need diversity because it's important. So the common misconceptions. So just a few here. So saying I'm probably not eligible to participate in a clinical trial, or trials are only for those at the end of the road and have no other treatment options available. And I remember speaking with one of my patients about mentioning a clinical trial. She started bawling and thinking that was the end. That means I have no other options and this is the end. And that's absolutely not the case. Usually with a clinical trial, you have to be robust enough to participate, and that's again part of the inclusion criteria. And what I say is it could potentially allow you an opportunity to get a treatment well before it gets approved. The other thing is, if my doctor is talking to me about a trial, that means I'm really not doing well and I have no other options. So again, that's something that is a myth or a misconception, or I'll be treated like a guinea pig. So really understanding that the goal of the treatment is to, yes, understand the drug, and it is something that will contribute to the future, but it may allow one to have access to a therapy that may be very effective for treating their cancer, or more safe for treating their cancer. And there's very diligent, evaluation prior to the study is kind of what I tried to lay out briefly today. I will get a placebo or a sugar pill instead of an actual treatment. So that's not ethical. So if there is an alternative therapy and there's a placebo there, it will not be placebo only. It may be that it's a standard treatment versus adding a new therapy versus a standard treatment plus a placebo. But you wouldn't have no treatment in an arm. There are certain types of conditions where there is no available treatment. And so what they're trying to do is here continuing usual care. So for example, supportive care, whatnot, versus a treatment. And so in that situation, it may be possible, but it will always be very, very clear. And that's in situations that would be quite unusual for breast cancer. It would be situations where we don't have any known approved treatments. So if I start a trial, I can never leave it. So that's another misconception. So I do tell my patients when we talk about trials is it's kind of like you're signing a marriage contract, right? So you're not going in with the next day planning to divorce it, but you can divorce down the line. Or I could say we need to divorce it, right? So if it's something that's causing more harm than benefit for one, then those are things. Or if it's like I can't do this anymore, whatever, it may be, you don't have to continue on the trial. What we don't want is we don't want people to like enroll and then they find out they don't get the treatment that they wanted and then they pull out of it. Sometimes you know what the treatment is, sometimes you don't, but that really actually impacts the statistics of the study and can be a problem. So it's really important that if you're going to consider it, that you really think about it, you mull it over with your doctor and your family and your loved ones, because it is something that it requires some level of commitment. Another one is it'll be too expensive to participate in a clinical trial. And so clinical trials do require some level of time commitment and driving back and forth and lodging if you live a distance away. So it certainly does that, but there are a number of studies that do also provide access to the treatment. So even if it's a standard treatment, they may provide it free of cost. And I think for me, that's certainly valuable to be able to get access to a drug because we know how expensive medications are. Or many studies will also allow for mileage or lodging or gas reimbursement, or they'll give vouchers. So talking to your doctor and the study team about some of those details. The other one is my oncologist will tell me if I'm eligible for participation in a trial. I think it's important for people to ask about this because it may not necessarily be the case, and then understanding why a trial may not be the right option for me at the moment. We usually don't recommend, and I don't, many of us would agree it wouldn't be ethical to have a very good standard treatment and then to then go to a very early drug when we have a lot of good standard treatments during the phase where the treatment is very, very early. So it's really understanding what's the right time for a trial and how is it going to be in my best interest. Even though a lot of it is volunteer and you're giving back, and I think it's incredibly it's something that I think is incredibly inspiring when I see those that are participating in research and giving back. And but it's also the goal is, what's going to be in it for me and how am I going to do with this treatment. And then the last one is it won't make a difference whether I participate or not. And so I have said again and again that this is voluntary, it's reliant on volunteers, and this is the way that we're going to advance science and medical care for everybody. So it will actually make a difference. So the last couple of slides is related to clinical trial initiatives regarding medication dosing and side effects. So we have Project Optimus. So this is a project that's through the FDA. And the goal here is to change the drug dose finding paradigm. So right now, what we've done over the years is we identify a drug and then we test different doses and then we find a dose and move it forward. But the goal now has been for a number of years is to not just identify one dose, but perhaps we don't have to treat it the highest dose where we have, we've maxed out on potential risk of side effects. Perhaps we can get a good biologic dose at a lower dose and really minimize side effects. And we're understanding that we don't always have to be at the highest dose. And your doctor may tell you, it's okay for us to dial down, because what we've done over time is we've often gone to the very highest dose, what we call the maximum tolerated dose, and then that's been the dose that gets approved eventually and gets developed. But we realize that we can still be biologically active with that treatment, even with a lower dose, and then you would have less side effects. So it's really identifying different doses that might move forward rather than one dose that you're going to study. And the goal here is to maximize efficacy, but also tolerability and safety so that patients can better tolerate treatments. So I kind of described this, but it's really, here this is kind of currently what we use. So maximum tolerated dose, where we start one dose level, we enroll a few patients, we see how they do, then we go up to a higher dose. If they're doing okay, we go up to a higher dose. If they're doing okay, we go up to a higher dose. And then we say, okay, now we have, too many side effects. We're going to go a little bit below that and then call that, and then, enroll more patients and move that forward. But what we're trying to do now is dose optimization under this new approach where we're looking at various doses, and then based off of those PKs, based off of those blood collections that we talked about and looking at levels, maybe we'll move on, a couple of doses and then identify the optimal dose. Again, that's based off of the blood tests and the different testing that's done. The other one that this is patient-led. This is the patient-centered dosing initiative. This is through the MBC Alliance. So they conducted a survey of thousands of patients with metastatic breast cancer and oncologists, and 86% of patients voiced that they experienced one or more bad side effects from their treatment. And 1 out of 5 of these patients visited a hospital due to side effects. 85% of oncologists do not believe that higher doses are always more effective. So this is feedback from the survey. And 97% of oncologists would be willing to explore flexible dosing with patients. And so this led to this initiative where it's understanding that we don't have to go to that top dose, and really understanding how to better design clinical trials from the beginning. So we're selecting that dose that's the right dose from an efficacy perspective and a side effect perspective. Okay, I'm going to stop there. Did I stop share?
No, you're currently sharing still.
Am I still sharing? Okay, let me see. Sorry. Okay.
Good. Yes. Say if Dr. Ma, if you want to jump in with your slides, then we can.
Questions in the chat, but we have more time to discuss later. So,
Yes.
Okay, let me share my screen. So I will, start the second part of this webinar. So thanks, Dr. Bagegni, for the comprehensive overview of, participating in clinical trial, what that means for patients, and the various phases of the trial. Welcome everyone, for the webinar. So I'm a medical oncologist, and I also see patients every day with breast cancer. So my goal today is, rather than talking to you about specific trials, I thought maybe it would be a good idea for you to kind of have a basic understanding what we have in the clinic now available to treat metastatic breast cancer, and what are potential exciting new treatments that are being studied in clinical trials, including a lot of them already in Siteman Cancer Center. And also, why trials are such an important part of patient care. And as Dr. Bagegni mentioned earlier, that clinical trials are really not a last resort. A lot of the times, these are really the opportunity to get on to the best treatment first become available in the trials. So I hope by the end of the talk, you'll be feeling more informed, and hopefully this will be helpful. So I think, one thing I like to show you is that, these are what we call Kaplan-Meier curves. So, kind of a positive note that over the past few decades, the outcome for patients with metastatic breast cancer has really improved significantly. So here on these 4 graphs you can see, so we have hormone receptor positive, HER2 negative, hormone receptor positive, HER2 positive patients, hormone receptor negative, HER2 positive, or triple negative. So all subtypes over time. You can see on the y-axis, this is looking at median survival after metastasis. And look that this is ranging from looking from 2000 to 2019, and now we're already 2025. I don't have that data yet, but it's pretty, we're pretty sure that, the curve continued to go up. And so this is a positive note that, with research and clinical trials, patients are living longer and hopefully also with better quality of life. And in many cases of, we know and our patient start to understand that metastatic breast cancer is being managed like a chronic condition, although over time we may have to keep changing the medications, and a lot of times clinical trials may play a part in the care. And just looking at these improvements, these do not just happen by accident, and they're really the direct results of advances in treatments. So, Dr. Bagegni showed you one slide earlier that, how the treatment has evolved. So many different drugs have been approved recently, and that really kind of evolving over time. We have better chemotherapy, hormonal therapy, target therapy, and I will talk to you a little bit about the antibody drug conjugates, kind of the new targeted chemotherapy drugs and potential immunotherapies. And so every one of these advances would come from clinical trials. So also I wanted to make sure that just to reiterate from Dr. Bagegni's discussion that clinical trial doesn't mean that there are no good options. It's just that there may be better options for patients. So I think to understand the different therapeutics or what's in the pipelines, more exciting drugs, I think would be helpful to understand, why they're cancer cells, what are the properties of cancer cells. So, if you look at the left side, a normal cell, they're pretty, normal, regular, they only grow when the body needs it. For example, if you get a cut, your skin needs to kind of proliferate. They need to grow, to heal, and then renew. And then when it's healed, the cell should not be continue to grow. And actually, they would stop dividing when the job is done. And they respond to the signals from the neighboring cells. So they should not be invading through, going to the blood vessels, kind of circulating your blood, for example, and they should not be metastasized. And actually, in healthy adult, I think it's good to know that only limited to tissues such as bone marrows or hair follicles or skin or mucosal cells are constantly dividing, to meet the needs that the blood cells are not immortal, and also the hairs keep growing, and you have new skins or mucosal cells, kind of replenish the older ones. So, but majority of the cells in your body actually are not dividing. For example, in adult people, I guess, your kidney is just that the same size, your liver is the same size, kind of it's not growing. But cancer cells, they would break these rules. Here, I show you the hallmark of cancer. What it's saying is that they would continue to grow, kind of, they have sustained proliferation signal. So because they have mutated the genes that's important to control the cell divisions, for example, or like they have the oncogenes being mutated, that they don't need to listen to the neighboring cells or growth factors, and they just grow uncontrollably. And also they can now spread to other parts of the body, like called invasion and metastasis. In addition, they may evade your immune cells. Supposedly your body has all these immune cells, like for example, T cells, B cells, plasma cells, macrophages, or other NK cells. All these cells supposed to be able to recognize foreign bodies or foreign cells. Like for example, when you get infection, these cells will go there and attack. And when you have cancer cells, they should have, they should be abnormal, and your body should be recognizing. But because cancer cells develop their mechanisms not to be recognized, so they evade immune response, and they attract blood vessels called angiogenesis. So, the reason I want to talk to you about these is because this is, these understanding looking at differences between normal cells and cancer cells, allowed researchers cancer cells and not normal cells. So, the treatment can be effective, but also safe. So also, one thing to remember is that cancer cells, they do not exist in isolation. Here, I show you this surrounding, the tumor cells actually is surrounded by a microenvironment composed of all different other cells like blood vessels, immune cells, and supportive tissues. And some of these cells actually are immune suppressive. Like, for example, they would inhibit your immune cells to attack the cancer cells. So, and they're called immune suppressive microenvironment. Sometimes, like macrophages, for example, may be exerting that way. And so there are drugs that not only are targeting the cancer cells, but also trying to change the immune microenvironment, making it, more effective in recognizing cancer cells. So, kind of, however, though, the current treatment we have, we are actually only limited in certain aspects or targets. And that's kind of where all the clinical trials come in. So here I want to just give you kind of a brief overview about chemotherapy. These are the oldest drugs that we have. So essentially, they're drugs that are really attacking, cancer cells or cells that are dividing. So mostly, they are not very cancer cell specific. But because cancer cells tend to divide fast, so they tend to be, chemotherapy drugs tend to be more effective for cancer cells. So, you can see on the top, I did show you here, there are various different type of chemotherapies, the DNA alkylating agents. So we have a few of them. They go to the nucleus and then bind to the DNA and cause DNA damage. And that's how they kill the cancer cells. And then we have like what they call antimetabolites. Essentially, they're the building blocks for DNA replication. So essentially, in order for cells to divide, you have to double the DNA contents. So these drugs works in that way. And then we also have like anthracyclines, and these can cause free radicals and interactions with DNA as well to cause the DNA damage. And then we have drugs that are called antimitotics. Essentially, in order for cells to divide from 1 to 2, you need these microtubules to drag these chromosome, into the 2 different daughter cells. But like microtubules are really essential in that process. So, these antimitotic drugs be able to, block that process. But all these drugs works better in dividing cells, but also they cause side effects. So for example, hair loss, because these are the cells that are actually actively dividing, as well as, gastrointestinal side effects, decreased blood counts, and that's because your bone marrows can be affected. So these cells these treatments are really the older generation of drugs, but they are still used in our patients when targeted therapies are not or no longer available. There are also newer chemotherapy drugs trying to really what they call antibody drug conjugate. So what they do is, these antibodies would take these chemotherapy, what they call payload. They link, kind of the antibody is linked to the payload, which is chemotherapy. They attach these chemotherapy drugs to the antibody, so they're kind of the targeted treatment to the cancer cell. The antibody would recognize cancer cells specifically. So these are, we have several different antibody drug conjugates. So for example, you may be familiar with Enhertu called trastuzumab deruxtecan or Dato-DXd. This is another drug that was recently approved, and then the sacituzumab govitecan, but they all use similar kind of chemotherapy drug called topoisomerase 1 inhibitor. very similarly, they cause DNA damage, and that's how they work. But then we also have microtubule drug as the payload, which is TDM1. So, but as you can see, we have very limited targeted chemotherapy. So, so one of the most exciting area in drug development for cancer is to develop these smart chemotherapy drugs. So they, so not just limited to, like, for example, HER2 or TRP2, those are the two antigen that we have that these antibodies target, but also, trying to get additional chemotherapy class. And then, in addition to the chemotherapy that I just talked to you about, so there are targeted therapies for, especially for estrogen receptor positive, HER2 negative breast cancer. We have more options because of the, recognizing estrogen is the major driver for the estrogen receptor positive breast cancer. So thinking about, if this is a membrane, and then like cellular membrane, and then you have estrogen in the blood, it will go binds to the estrogen receptor called ER, to the cancer cell, and then eventually it would turn on the cell cycle, and then kind of move the cells to cell division. But so we have a lot of drugs that would reduce estrogen levels. So, for example, those aromatase inhibitors or drugs that would modulate estrogen receptor. But also more recently, we have, two drugs FDA approved, elacestrant and imlunestrant. They would target the mutated estrogen receptor that happens, when cancer is exposed to like decreased estrogen for a long time. Now they don't need estrogen anymore, so they just need that mutation. So now we have these drugs. And in addition, there are so what we call growth factor receptor pathways. So in addition to the estrogen receptor pathway, scientists recognize that these intracellular molecules that mediates the growth factor, what essentially is an independent pathways, and especially mutations in key kinases, PI3K, AKT, and mTOR, they can cause resistance to endocrine therapy. And that led to all these approvals of PI3 kinase inhibitor, AKT inhibitor, everolimus. But there are a lot of side effects associated with this drug. And so a lot of the effort is being developed to target specific mutations related to those drugs. And then, a lot of you probably know about these CDK4/6 inhibitors. They're really downstream of all these growth factor pathway or estrogen receptor pathway. So they're in the cell cycle, essentially prevent the cells from, making the DNA for them to proliferate. So essentially these are, available, drugs for the ER positive breast cancer. For HER2 positives, HER2 essentially is on the surface of the cells. HER2 positive cancer essentially they rely on that HER2 pathway and is overexpressed. So we have a lot of antibodies, and we have chemotherapy targeted chemotherapy, the antibody drug conjugates, and we have HER2 inhibitors. And so there are a lot of drug development in these areas trying to make more effective HER2 inhibitor that able to penetrate the blood-brain barrier, kind of be able to treat metastasis in the brain. And then we have things like drugs that target specific mutations, not specific for breast cancer, can be any other cancers. And then the immunotherapy, which is currently only available for triple negative breast cancer, because these cancers tend to have more immune cell infiltration. So they need, once you give these immunotherapy that, the cancer cells can respond. So essentially what these immunotherapy do is, specifically for this particular PD-1 inhibitor is that, so these are called immune checkpoint, the LAG3, PD-1, and others. So they think about this is on the left side is it immune cell. Here is a T cell. A lot of times is really the soldier kind of for the immune cells. Then this PD-1 expressed on the immune cells can be recognized by tumor cells through this PD-L1, which is the ligand. So, and that's how tumor cells neutralize the T cells, telling them, where we are, normal. Because normal cells use these immune checkpoint, PD-L1, so that, our T cells are not really recognizing ourselves, not causing any immune autoimmune disease. So, but when you give these antibodies, now you dissociate that interaction. Now the immune cells can recognize tumor cells. So, but these are all, everything that we have. As you can see that, there can be a lot of resistant mechanism. Cancer cells are so much more complicated than we have here to be able to, overcome all the treatments. And here I thought would be interesting to talk to you about PARP inhibitor. You don't have to really understand this whole thing. Essentially, this is specific for patients with BRCA1 or 2 mutations. Think about BRCA1 and 2 is important for DNA repair, but then there are other pathways that needed for DNA damage repair, and these are where the PARPs are. So when you inhibit PARP, then the cells accumulate a lot of DNA damage due to the inability of the BRCA to work or the PARP to work. So they call it synthetic lethal. Essentially, these olaparib, talazoparib, and how that works. So the reason I think, talk to you about this one is because, so earlier we talked about, how to attack cancer cells through, using chemotherapy or using target therapy directly, kind of inhibit the driver of the cell growth signaling. But here really is kind of taking advantage of, the tumor cells are, lost of a tumor suppressor. Now we can use a different way to treat the cancer. So there are a lot of drugs that are trying to inhibit the DNA damage repair pathways. And those inhibitors, the thought is so that we don't limit to BRCA mutated tumors. So these are, something that I'm going to highlight. So, kind of that's, I hope it's not too complicated, but I thought maybe helpful for you to understand. So the evolving cancer therapeutics that I want to highlight just some of the strategies being developed in the clinical trials. A major focus is how to improve our portfolio for the antibody drug conjugates, making it more effective and safer. And then for ER-positive breast cancer, resistance to CDK4/6 inhibitor, which is our first line, treatments is a big problem because after patient progress on those, the treatment that we used to have, they're not as good, but, there are newer drugs being developed. So I think there, a lot of excitement on novel targeted therapy. And then I think we're all excited about immunotherapy because they offer the hope of cure because, mobilizing your own immune system is the best way to treat the cancer. And then, I'd like to just highlight, just use one slide to talk about DNA damage response pathway inhibitors. So, I think, the antibody drug conjugate, just to tell you a little bit more about how it works. This antibody, so it has this, what it does is recognize specific proteins. And then there's a linker that can link to this, what they call cytotoxic payload, essentially chemotherapy. So this antibody would recognize antigens or cell surface proteins on the tumor cell, and then bring the chemotherapy to the cancer cell. And so this diagram just shows you the kind of the key features, how it works. Think about the red dots are chemotherapy, and this antibody recognizes this purple dot, internalized by the tumor cell. And then in the tumor cell, they will release these chemotherapy. Actually, these are the chemotherapy that going to cause cell death of the cancer cell. They may be, causing inhibition of the microtubules, or they may disrupt the DNA, DNA, causing DNA damage. So these are the main classes of the cytotoxic drug that we have for now. But, we still have limited numbers available drugs. So, what's in the clinical trials or in the pipelines are now these, so they're trying to design the antibody drug conjugate by modifying the target. So where the antibody recognizes. And these are all the new potential targets that the clinical trials or most of these are drug companies that they're using these novel targets. Essentially, these targets are the ones that are overexpressed in cancer cells, but not in normal cells. So this way you can allow the chemotherapy to be targeted specifically. So I highlighted on the right side, those are the few, kind of the antigens or antibody targets being, designed using, like, for example, HER3 antibody drug conjugate, Nectin-4, B7H3, or tissue factors. These are the things being, tested. And then some antibody drug conjugates, what they're trying to do is rather recognizing one antibody recognizing one antigen, like just HER2, for example, they're also going to recognize TRP2. Kind of now you have two surface, kind of two proteins on the surface of the tumor cell that increases the affinity of the antibody drug conjugate to the cancer cell, but also more likelihood that this drug may work because the cancer cell may have HER2 or it may have TRP2, for example. And in this small trial, so the really early phase trial, so they had a response rate of 31 to 44%, meaning that about over a third of the patient had their tumor shrinkage over 30% of their tumor, shrink. I think that's really impressive. And a lot of them are kind of hormone receptor positive, triple negative. And this is a bispecific antibody that recognize two surface proteins. And then sometimes they would design this antibody to recognize different part of the same protein. And in this case, they call biparatopic. And this are recognizing HER2 for this particular antibody drug conjugates. And this is in HER2 positive cancer. Response rate was 75%. So those are kind of having already very early signals. So, these are all phase 1 trials, actually. So with phase 1 trial, actually, a lot of the responses already seen. And there are also newer drugs trying to, using two different chemotherapy drugs to be attached to these antibody. So this is an animal study, but what you needed to focus on is this this blue curve. So looking at tumor growth over time, when you give this antibody drug conjugates, the tumor is not growing. But compared to if you give the two chemotherapy antibody drug conjugates separately, then the tumor is still growing. So these drugs are also in development. So rather than attaching one type of chemo, you can attach more chemotherapy. And then you can also attach like what they call immune modulators. So essentially, so here, for example, they attach this, what they call TLR7/8 agonist. Essentially, this is important to stimulate immune response. And people have seen really good tumor response as well. So these antibody drug conjugates have various ways to design, and sometimes they would put radioisotope, so to the rather than a chemotherapy. And these are also in clinical trials. So bring in radiation specifically to metastatic cancer cells. And this may be a little difficult to explain, but it's just saying that, over the years, we have multiple different chemotherapy drugs are being, trying to be tested and designed into the antibody drug conjugates. So now there are over 200 antibody drug conjugates in clinical development. So, diversify the chemotherapy component of it is a big part of these trials. And here are the various different targets or different chemotherapy part or targeted therapy. They're trying to attach these as well, to these antibody drug conjugates. So, in our phase 1 group, they have multiple antibody drug conjugates with various properties being tested. And so next, I'll just focus a little bit on the CDK4/6 inhibitor resistance. I will only just show you one slide. So, to be able to be smart at how to treat patients with tumor cells, already resistant to CDK4/6 inhibitors, you have to understand, what are the new things or genomics that drive these tumor growth. So, on the right side, so this is what, clinical investigators, when they looked at patients' samples, after the patient progressed on CDK4/6 inhibitor, what they found was in the blood, what they call circulating tumor DNA, you can see, these, or tumors, you can see these aurora kinases, which is important for, controlling the cell division, or cyclin E, which is also can drive cell cycles, and HER2, for example, all these different molecules, mutations or amplifications or loss of them. So, and that's how the different inhibitors, and some of these are fairly, established targets already. So, for example, people are developing CDK2 inhibitors because this is a pathway that can have cancer cell, after cancer cell escape to the CDK4/6 inhibitor. Now they rely on CDK2. And then, there are other, transcriptional CDKs. So they're, the hope is that these target drugs be able to, provide additional, benefit, and then kind of epigenetic modulators and upstream kind of the growth factor inhibitors, and then other, intermediate kinases that mediate the, tumor, kind of the oncogenic signaling pathways. So in addition, to the PI3 kinase pathway, but there are also like MAP kinase pathways. So these are, this is a very brief kind of an outline of what are most commonly tested in clinical trials. So at WashU here, we have several trials that target CDK2. They're CDK2 inhibitors. There are also trials with aurora kinase inhibitors, KAT6 inhibitors. So these are all in the breast cancer portfolio, and FGFR HER2 inhibitors. So these are all available. And of course, there are, this is not comprehensive, but just to kind of give you a flavor, what are the, the rationales for these studies. And then for novel immunotherapies, if you look at on the right side, we talked about immune checkpoint. We have PD-1 inhibitor, but this is only one of the several checkpoints that we know of. There are other checkpoints. So there are potential combination therapies that are being developed, trying to, dissociate all these different checkpoints. And then there are, for example, cellular therapies. In breast cancer, we're still very, very early in cellular therapies, but there, investigators are trying to develop these tumor-infiltrating lymphocytes therapy. So essentially, you isolate the tumor lymphocytes, and then you expand them outside of the patient, ex vivo, and they infuse back. So that's being tested. We do not have that yet here at WashU, but those are in development. And then there are what they call CAR T cell, like chimeric antigen receptor T cells. essentially, you also get the T cell lymphocytes from patients, but then you engineer them so that they now can recognize cancer cells and you expand them. And some of the tumor types already have CAR T cells like lymphoma, but for breast cancer, we do not have that yet. But these are things potentially will be coming. WashU is very good with cancer vaccine studies. So essentially, these vaccines are made based on mutations in the cancer cell. So you have to go through these genomic sequencing of the cancer cell and then determine which particular mutations may make the cancer cell respond to immunotherapy and then give to the patient as like a vaccine. So these are all in development, but overall immunotherapies are still very early. And then, for the DNA damage response pathway inhibitors, so on the left side here, there are many of these drugs in the clinic. So the reason is, kind of the DNA damage repair. So on the top here, on the right side, you can see the DNA double strand. But when it gets single strand break or double strand break due to, for example, when you get chemotherapy, you may have that. But how can we use these inhibitors to potentiate, these chemotherapy effect or combining different DNA damage repair pathway inhibitors? So these are also, they're a big focus of Siteman is to, there's we have a genomic integrity center, so institute actually called Gene Scientists trying to work on what are the most critical targets and how to inhibit that. And then in our phase 1 group and as well as our breast group, we have several trials that are targeting different ones. For example, of course, the PARP, now they're, getting more selective PARP inhibitors, PARG inhibitors, and then Pol theta, WEE1, all these inhibitors are also in the clinical trials here. And then some drugs are potentially more effective for tumors with certain particular mutations. So, but although these are still being studied. So I think, to summarize, these clinical trials really drive progress, and that's because of improved understanding of the cancer biology. Actually, we study a lot from our patients. You may patients a lot of times are being approached for what we call, research studies of, for example, tumor banking studies. So looking at collecting patient tumor specimens or blood to understand why the patient progressed on the current treatment. And that's very important. And also at Washington University here, we have a program called patient-derived xenograft models. So what we would do is, getting the patient samples into the lab so that these would either grow in the petri dish or they will grow in immunocompromised, mouse models so that these can be used to study biology. So, what I told you earlier, just these are still evolving, but I thought these are among the most exciting new drugs. Hopefully for the next few years, we would see some new approvals. So that's all. Thank you. Thank you for your attention.